Okay, I think we'll go ahead and get started. Welcome, everybody. It's a pleasure to welcome you back to Convocation Hall. Today, it's an extreme pleasure of myself and Saman to welcome Doerthe Tetzlaff from the University of Aberdeen. Before I introduce her, I just want to thank, as always, Howard Weider and the Canada Excellence Research Chair and the Global Institute for Water Security for underwriting this seminar series. And to remind you that next week we have Chris Soulsby from University of Aberdeen who will be speaking here. So today we have Doerthe Tetzlaff. She's professor and chair in hydrology and landscape ecology at the University of Aberdeen. She completed her PhD in hydrology at the Institute for Hydrology at the University of Freiburg. That was in 2004. And then following a 2-year postdoc at Aberdeen, she served there in faculty capacity since then, becoming a full professor back a few years ago now, which is quite remarkable given her tender age. She currently leads a very large and prestigious 5-year European Research Council grant, some of which you'll hear about today, and this is focused on outstanding young researchers. And not surprisingly, she's been awarded several honors in her career thus far: a Doctor of Science from University of Aberdeen. She's a member of the Royal Society of Edinburgh, Young Academy of Scotland, and a visiting fellowship in freshwater science from the Leibniz Institute. Doerthe's had a lot of professional leadership roles in her career. She's served as editor of Hydrology and Water Resource section of the Geography Compass. She served as editorial board member of Hydrological Processes starting in 2010, and in 2015 rose to the position of editor-in-chief, where she now is really the one at the helm of hydrological processes, and I know a lot of the students and postdocs have been chatting with her about the journal this week. She does a lot of work for AGU that I'm so appreciative of. She's chair of the AGU Hydrology Section Surface Water Technical Committee. She's served on the AGU Early Career Award Committee, and has had quite a significant involvement with PUB, leading one of the themes some years ago when that was a major activity of IHS. Of course, she's been a convener of many, many sessions at AGU, EGU, and co-led the Gordon Research Conference on Catchment Science. So it's really a pleasure to have Doerthe here tell us about her take on catchment science, and in particular advances in ecohydrology in changing northern regions. Doerthe.
Okay, yeah, so the microphone is on. It's really a pleasure to be here, not just because it took me about 48 hours in transit to reach Saskatoon. This was not due to the distance, but really to weather situation back in Germany. But it's also a real honor to be here, mainly being part of this speaker series of distinct speakers you had here the last few years, but also being able to visit the Global Institute. I think it is clear in recent years, it's not just a name, Global Institute, it's really globally known. And I remember a few years ago, I went to the CGU conference in Banff and was just astonished that almost every second speaker seemed to have come here from Saskatoon, so that's very impressive. And I would like to use the opportunity today to talk about the work of our group in Aberdeen in Scotland and how we try to advance our process understanding of ecohydrological processes in changing northern regions. So the work I'm talking about today wouldn't have been possible on my own, and hence I would like to thank some key people here first. That's Chris Soulsby, who will talk here next week in giving a seminar. We also were in a fortunate position to work with really great, clever and hardworking postdocs over the past few years who are listed here. And then I've been in the fortunate position to work with international collaborators who are listed here as well. They were very inspiring over the past few years. We have the local stars, of course, Jeff and Chris Spence, and also many others who I would like to thank here first before I start. Probably everyone, most of you in the audience knows when we try to manage ecosystems in a sustainable way, we need to, and for human use and as a living entity, we need to consider that aquatic and terrestrial ecosystems are coupled at multiple scales. So this can range from interactions at the reach scale, then moving towards the larger river scale, when we think, for example, about hydropower implications, and then the catchment scale, thinking about land use implication and the consequent interactions. And first, it requires a fundamental process understanding of the in-stream ecological functioning to inform stakeholders and for sustainable management. And some of you might know that our group in Aberdeen has done a lot of work on in-stream ecology, but in my talk today, I won't talk about this in-stream ecology work at all, but I just wanted to mention it again. The focus of my talk is are really the terrestrial ecosystems. And we know that what's happening in our rivers, in our freshwater ecosystems, is strongly influenced by what's happening in the terrestrial ecosystems. So we need to understand the processes in the landscape, and to do this, we try to answer seemingly simple questions, which are listed here, but they are quite difficult to understand and to investigate. So that's really, where is water stored in the landscape? What are the fluxes out of our catchment? What are the ages of these fluxes? And how do these processes affect the water quality and in-stream ecology? And I know that many of you here in the audience are working on some or if not all of these questions as well. In recent years, our group focused more trying on the understanding of the role of vegetation in the terms of partitioning, the release, and the movement of waters in our landscape. And again, I know that's the focus for many groups and also many colleagues here in Saskatoon. In Europe, probably more than here, I need to emphasize that snow-influenced northern ecosystems, where most of my work is based on, are very sensitive to climate change because, as we know, even small temperature changes determine whether precipitation falls as snow or rain. They affect winter snowpack accumulation and the rate of melt. In addition, these northern systems also experience quite specific land use pressures. So here are some examples shown from the Scottish Highlands or the Scottish upland areas, but they apply to many northern ecosystems. So for example, we have the expansion of renewable energy, the expansion of wind farms, we have the use of hydropower. We can experience afforestation efforts or changes in crop rotation when we think about the expansion of biofuel crops, and then deforestation. And all these climate and land use pressures have, of course, important implications, not just on the stream flow regime and the water quality in these systems, but also on the ecohydrology, both in terms of in-stream and terrestrial ecohydrology. We know that vegetation communities are responding to a warming climate in terms of changing their composition and their distribution, and indeed in many parts of the wider north we do see these vegetation changes already occurring. So for example, in terms of an increase in the shrub or tree cover through these warming temperatures. But the subsequent effects of these climate-induced vegetation changes on evapotranspiration or soil properties are still largely unknown in terms of altering the precipitation input and the partitioning of precipitation and the changes of subsequent storage and fluxes of water. And these questions have been to the core already of a previous international site comparison study, which I was leading a few years ago, which was called Northwatch. And part of Northwatch were 10 iconic long-term experimental sites across the wider north. We had in the Scottish, 3 sites in Scotland, the Strontium, the Gurnick site, and the Altemarki site. We had a site in northern Sweden, the Crickland site. We had 3 sites here in Canada. I have to see the arrow of the Wolf Creek has moved there a bit, but we had the Wolf Creek site in the Yukon Territory. We had Catamaran Brook and Dorset. And then also in the US, we had the H.E. Andrews sites, Hobart Brook and Sleepers River. And for these sites, we had long-term data of hydroclimate variables, but also of biogeochemical data and ecohydrology data. We have done all kinds of analysis with these data set, and if you are interested, I would like to refer you to my webpage. But in the context of today's talk, one of the analysis we have done was applying quite a simple biome classification system using mean annual temperatures and mean annual precipitation values for our 10 sites. And even if simple, we found that this classification system was really a useful systematic basis helping us to conceptualize possible future changes in the ecohydrology. So what we see in this plot, which looks a bit complicated but actually isn't, is in black are the dots for our 10 sites classified for current mean annual temperatures and current mean annual precipitation values, and then plotted in their current biomes, if you want. And then the white dots show the shift or the change in mean annual temperatures and mean annual precipitations based on current climate change projections. And we found that at least at some of the sites, some climate change projection might reside in a shift of the biome. And we also found that these impacts seem to be largest at the sites furthest north of our site comparison study. So these sites in the north seem to be catchment on the cusp of change, and this phrase was coined by our colleague Jim Buttle. So for example, when we use our site in the Yukon Territory in Canada called Wolf's Creek WC here, it seems due to the climate change projections, we might experience a change from a current tundra biome system towards a more boreal forest system due to that the warming temperatures are affecting the disposition of the forest through this upward migration of the tree line. Another example is KR, that's our site in northern Sweden. Again, a possible shift from a current boreal forest system towards a more mixed forest system. But what is still unknown is whether to which extent these species adapt to climate change, and what's also unknown, how such possible vegetation changes might affect the storage and the release of water in our systems. And hence these questions are also to the core of the current international project which I'm leading and Jeff was mentioning briefly, which is called VeWa, where we are trying to understand these vegetation, soil, water interlinkages and how they might be affected by climate change. So core of VeWa are again 6 long-term experimental upland catchments across the north. They are a subset of the original Northwatch site. And important is that the sites plot along a hydroclimatic gradient in terms of their snowiness and their seasonality in water replenishment. So we have again a site in Scotland that's a tributary of the Gurnick site, which I was mentioning earlier. We have our Crickland site in northern Sweden. Again, 3 sites in Canada, Wolf Creek, Yukon Territory, Moss Creek in Northern Territory, and the Dorset and Ontario, and also 1 site in the US called Dry Creek. And here on this panel, you hopefully get an idea about the hydroclimatic spectrum we are covering with these sites. We are plotted here several years of hydroclimatic data in terms of mean annual temperatures, mean annual precipitations, just really to give you an idea about the spectrum the sites cover. In my talk today, I will talk or mainly focus on the Scottish site, the Bruntland Burn, and give you examples. Important is the Bruntland Burn is one of the least snowy sites of all the VeWa sites, but we are applying the same sampling strategies across all the 6 VeWa sites. And important is also to keep in mind throughout my talk that the models we are developing and testing in the Bruntland Burn are meant to be applied across all these 6 VeWa sites. So the Bruntland Burn is typical for many upland ecosystems in northern Europe in terms of being an old glaciated landscape. We have quite a subdued topography. The annual precipitation is about 1,100 millimeter. But important is, again, to keep in mind we have a very limited seasonality, so precipitation is evenly distributed throughout the year. As I just said, snow plays a minor role of just about 5% of annual precipitation, and we have a low PT rates of just about 400 millimeter. I hope you can see on this background picture already that we can identify distinct vegetation soil units in the catchment. So in these wide valleys, we have these extensive sphagnum-dominated peatlands, and you see this here with this light green vegetation cover. So these are extensive peatlands and wetland areas. You also get an impression of these photographed during a different time of the year, but just how much water can stand in these systems. They develop on deep organic soils. They are histosoils, which can be up to 4 meters deep and are highly water retentive and hydrologically very responsive. The natural vegetation in Scotland would have been Scots pine forest, but due to really a long history of burning, of clearance and overgrazing by deer and sheep, now the dominant vegetation is this heather moorland vegetation, which looks a bit purplish here on this photograph. So we still have some stands of Scots pine. These are the stands where we have highest interception, highest evaporation rates, and I will get back to this later during my talk. But I also would like to point out already the quite open canopy of these Scots pine stands which we have. And both of these vegetation types, the moorland and the Scots pine, develop on freely draining pot soils which contribute to groundwater recharge. So as I said, within VeWa, we are trying to understand these linkages between the vegetation and the soils and how they are affecting the release, the movement, the storage of water. So in this graphic, we try to conceptualize the processes which we can actually measure relatively easy, and they are marked here in green, the physical processes which are relatively easy to measure. But often, as catchment scientists, what we try to understand are the processes which are marked here in red, and they are much more difficult to measure. It needs much more integrated, complex approaches to understand these processes. And within VeWa, it's really the stable isotopes which are a crucial tool we are using here in combination with other approaches to gain insights into this ecohydrological functioning of these systems. And I will give you more examples later during my talk. So crucial to VeWa is really that we combine stable isotopes measured in different waters in our systems with tracer-aided modeling approaches, and I will explain in a second what I mean by this. And we integrate these approaches to firstly investigate the storage, the flow paths, mixing and fluxes out of the system by using such coupled models of flows and tracers. We try to understand the vegetation effects on interception and precipitation partitioning. And then thirdly, we try to quantify the dynamics in soil-water isotopes in terms of evaporative losses and fractionation in these dominant vegetation soil units, which I was pointing out earlier. Crucial so is that the findings and insights we gain from these objectives 2 and 3 are feeding back into our objective 1. So the idea is that we use the data to improve the parameterization of our tracer-aided models. And to achieve these objectives, we are using integrated approaches across different spatial scales and across these different sites I was talking about. So we develop and apply tracer-aided models where we use the tracers to inform the model structure and to constrain the parameter sets. And we can then use such tracer-aided models to understand the routing of water and tracers through our landscape. Important is that these models are empirically based. This means we incorporate the isotopes we measure in the different stores or components of the models in terms of isotope measured in precipitation, in discharge, in the soils, and in the groundwater. But recently, these tracer-aided models have shown us that we still need much more detailed empirical field studies to really understand the role of vegetation on these hydrological processes, if you want. So for this, we recently focused our studies more on the plot and hillslope scale, and measuring also isotopes in throughfall, stem flow, and plant xylem water. And the idea of these plot and hillslope scale studies is really improving these parts of our models, which are important to incorporate the role of vegetation. So initially within VeWa, we conducted a simple meta-analysis simply to get to know our different sites and catchments. And we used the HPV rainfall runoff model to get an idea about differences in the partitioning of flows between soil and groundwater stores. So here are two examples shown. I've chosen our site in Scotland, which is one of our mildest and wettest sites from all the VeWa sites, and then the Wolf Creek site in the Yukon, one of our coldest and driest sites. And without wanting to go into any detail what we see here, I just want to emphasize the differences we see in the hydroclimate across these sites, and also how this is reflected and transferred in the differences in the storage dynamics and the partitioning of our flows. So each time on the upper panel, we have in blue our measured precipitation, and in black, we have our simulated snowpack accumulation. The middle panel shows the dynamics in our soil and groundwater stores, and then the lower panel shows the measured and simulated discharge. And I hope you can see already these differences. For example, at our Scottish site is evenly distributed precipitation input throughout the years. In black, we have some snowpack accumulation simulated, but it's of minor importance. We then have these large dynamics, particularly on the soil storage, and then these highly responsive hydrological response in our discharge. In contrast, the Wolf Creek, much more seasonal, the importance of snowpack much clearer, and also the seasonal response then in terms of the dynamic storage changes and the discharge response, really reflecting this importance of the snow melt at this site. And we can then contextualize these model results in these box plots, again for comparison purposes, and each time I marked in red our Scottish site as our mild, wet site, and then the Wolf Creek site as our coldest, driest site, or one of our coldest, driest site. Looking at the snowpack, you see this little importance of snow in Scotland, much more important, of course, at the Wolf Creek site. We also found that overall soil water storage was much larger, more important than the groundwater storage, if you consider the different scales at both plots. And this emphasizes already the importance of evaporation and transpiration processes in terms of determining the losses from the system. And we also found that the storage changes in the groundwater were the main driver of the simulated storm runoff response in the catchments. But of course, we acknowledge that such rainfall runoff modeling is a useful representation of the dynamic storage changes, but really representing the total storage in the system, the mixing processes, and the role of vegetation in tracer-aided models is much more difficult. So hence, in the second step, we look now at the stable isotope data of our site, and I forgot to mention this earlier, that actually this was a major rationale to incorporate experimental sites in our VeWa site comparison studies. We wanted long-term stable isotope data available to be able to contextualize our current sampling campaigns. So we were interested in, by just plotting our input-output signals, whether we can see any vegetation signal in these data. And what we see here is, this time I'm showing the Crickland site, in northern Sweden, which is our most northerly VeWa site. Again, the Wolf Creek site in the middle, and then our Scottish site on the lower panel. In black dots, we see our precipitation input signature, and characterized at all the sites by a high variability and a strong seasonality in the signal. And then the red dots show our stream water output signal, usually much more damped than our input signal due to mixing processes in the landscape, etc. And what we see are already some, again, subtle differences maybe, but helping us to understand the functioning of these systems. So we do see this depletion in our stream water response in response to snowmelt events at the Crickland site in northern Sweden. We also see that precipitation events in late summer or in autumn are important, influencing the isotopic response, but also for the discharge response. Wolf Creek, back then a very sparse data set, but I still wanted to show this here because it emphasizes really the difficulty in getting stable isotope data, any empirical data, in such remote environments like the Wolf Creek site. I have to emphasize that Sean Carey has done an amazing job over the past few years building up an almost consistent, continuous isotope data set for the Wolf Creek site. We are incorporating them now in the VeWa project, but in this initial metadata analysis, we didn't incorporate these data. But still, looking at the snowmelt period, and again, knowing about the importance at this site, we do see how the snowmelt is not just influencing the hydrological discharge response, but also influencing our isotope dynamics. And then our Scottish site, a very damped response in the stream water, emphasizing, telling us already that a lot of mixing is occurring at this site. We don't really see any snowmelt influence, and this large and consistent discharge response at the Scottish site. And then we use the stable isotope data to plot them in these dual isotope plots, and some of you, most of you might know that we can use such dual isotope plots to identify whether we have a deviation of our samples from what's called the meteoric waterline. So any change in slope or deviation from the meteoric waterline compared to the deviation of these water samples, whether they are measured in the soils, in the plant xylem water, in stream, lake, etc., tells us something about fractionation processes occurring and possible influence of the vegetation. So initially, we simply plotted our stream water isotopes we had available, again for all the sites, but I'm just showing here 3 of the sites. And in red are the samples taken during summer months in stream water, in blue are the samples taken during the winter months, and hopefully you can see not just the different scales here tells us something about how enriched or depleted our signal is, and I will get back to this later during my talk, but also the differences in the deviation from the meteoric waterline, telling us something about differences in the internal catchment processes on isotopic fractionation, on evaporation, in terms of, for example, the importance of snowmelt and snowpack, and also the percentage of wetland covers across these sites. But as I said earlier, we know this rainfall runoff modeling is just a simple partition, or just allows us a simple partitioning of the water. The next step was now trying to develop a tracer-aided, a spatial distributed tracer-aided model. And for this, in the first instance here, we see our input data set for our Scottish site, and we have now quite a unique data set available of almost 5 years of daily data in precipitation input, which is this time plotted in red, and stream water output, so this time plotted in black. Again, you see this dumping in the stream water signal, the high seasonality in our precipitation input signal, and we also have such time series available for isotopes in soil water and in groundwater. And we were then successful in incorporating these stable isotopes into this distributed tracer-aided model, which we called STARR. And Chris, in his talk next week, will talk much more about the model development and the findings of this model. But in the context of my talk, I just want to emphasize that this model still focuses on the terrestrial partitioning of water, and the role of vegetation is still quite limited within the model. So hence, in the last year, we really tried to focus on getting new data, getting new empirical data, focusing our studies more on the plot and hill slope scale to be able to improve the parametrization of our tracer-aided models. So in the following few slides, I would like now to give you examples for these processes which we try to measure and to understand, starting with measuring the amount and isotopic composition in precipitation, throughfall, and stem flow. We also conducted subflow measurements to understand the role and extent of tree transpiration. We put a lot of focus on trying to understand the soil evaporation and fractionation processes in our system. We started to incorporate these data already into a tracer-aided plot scale model, and the final overarching goal is to really develop a tracer-aided catchment scale ecohydrological model to integrate and combine all these different processes. So as I said, now in the following, I would like to give you examples for each of these different approaches. So starting with the throughfall measurements, we had at this site here 25 throughfall collectors available. And please remember what I said earlier during my talk, that these Scott spine stands, which we have in our site, are characterized by quite an open canopy. So on this graphic, we see in purple no canopy cover, and then in dark green, it's a dense canopy cover. The green triangles show where the trees are and the size of the trees, and then in orange, we have the average amount of throughfall sampled during the study period. And hopefully you can see that we found quite a marked spatial variation in our throughfall quantity. Overall, interception losses were about 40%, and throughfall comprised more than 99% of net precipitation, so stem flow was neglectable, and hence, during the talk, I won't refer to the stem flow any further. But despite this high variability in our throughfall amount, when we look at the isotopic signature on throughfall, we didn't really see any spatial variability. So again, this time the isotope signature is reflected by the size of the orange dots, again averaged over the study period. And you see in our ecosystem that's quite similar, telling us that in these parts of the system, not much fractionation occurs. And this is confirmed when we look here at this time series, where we have in pink dots a daily precipitation input signature, and the box plots show the throughfall signature at the weekly time scale. And I hope you can see how similar the signature of both input variables is, again confirming this, that not much fractionation occurs in these parts of the system. So we also conducted subflow measurements to understand forest transpiration, and this work was led by one of my postdocs, Hai-Long Wang. We see here the data from summer 2015. The data of 2016 are just analyzed while I speak. And we would expect highest transpiration rates in summer. So we see in the red line are the transpiration dynamics, the blue line is the averaged soil moisture with depths, and then the black columns are our precipitation input. And remember what I said earlier, we are in Scotland, so even in summer we have quite a lot of rainfall event, almost 2/3 of the days we do have some kind of rainfall, but important is that they fall in quite low intensity events of less than 5 millimeters per day. Looking at the soil moisture, we have this one peak of about 20% after this large rainfall, which was about 25 millimeters per day. Again, I have to emphasize such intensity events are quite rare in the Scottish context. But then, interestingly, despite this constant precipitation input, we have this decline in the soil moisture over summer to just about 15% or even less. Transpiration rates are quite low, are not very high. We are in a low energy environment. And as I said, this work is still in progress. So up to now, we can't really see whether this increase after these rainfall events is due to hydroclimatic conditions. On rainy days, we have quite cool, cloudy, humid conditions, so transpiration demands are low, or whether we do have, or whether the trees experience soil moisture stress and hence are responding immediately after rainfall events. So we are not clear about this yet. But following on from this, when we look at weekly water fluxes, we see here in blue columns the weekly precipitation amounts, and then the red line is the precipitation minus the transpiration as the transpiration demands. And we found that in most weeks the precipitation meets transpiration demands and is substantially higher than the transpiration. We do have some periods, but very limited, where we have a deficit, but this deficit is again quite limited, of less than 10 millimeters. And this, together with the almost declining soil moisture over summer, despite the rainfall, constant rainfall input, tells us that we seem to have a rapid uptake of water just from the upper soil horizon in these systems, and that a significant soil evaporation must occur during summer. And these findings are consistent with a pilot study we conducted a few years ago, where we did measure soil water isotopes and plant xylem isotopes. So again, you see here such dual isotope plot, which I was mentioning earlier, and all the little symbols are soil water samples measured in different depths and also using different extraction methods. But we see that for most of the year, the samples are plotting along the meteoric water line, showing no signs of fractionation. Looking at the plant samples, which are plotted in these big dots, again, it seems that for most of the years, they are isotopically similar to the soil water from the upper horizon, plotting along the meteoric water line. The only times when we do see some evidence of fractionation, so this means a deviation from the meteoric water line, is for these soil samples and for these plant samples is during dry summer months. And we think this is due to the soil evaporation just from the upper horizon of the soils. I have to emphasize so that this, when we did this pilot study, we measured in 10 centimeter depths and deeper. We know now that almost 85% of the fine roots of these trees are in the upper 10 centimeters. So it might be that in this pilot study, we haven't actually sampled the horizons where the trees are and the roots are getting their water from. So because of this, in the last year, we focused now our measurements just on these upper 20 centimeters of our soil horizons. And this work was led by one of my other postdocs, Matthias Sprenger. So Matthias sampled 5 locations across the site, covering these different vegetation soil units I was talking about earlier on a monthly basis, starting in September, October, so autumn, winter, spring, summer, back to autumn. And the plot is a bit complicated, so I just want to talk you through this gently. These are now just the 5 centimeter soil samples, but as I said, every 5 centimeters we have similar data. The color of the circles shows you which season we are in. The inner shading of the circle, and you will see this in a second a bit more, gives you an idea about the PET rates, the 30 days prior the sampling, with redder colors showing higher PET rates, bluer colors, low PET rates, and then the size of the stars gives you an idea about the weighted precipitation of the 14 days prior the sampling. So starting in September 2015, we see we are quite far away, plotting below the meteoric water line, which tells us during this time of the year we have sampled fractionated soil water. This is still the case even in October 2015. Also, our PET rates are getting now a bit bluer, so less PET rates. And then moving in November towards winter, now our soil water isotopic signatures become more depleted, and we are moving back to the meteoric water line. We didn't sample in December. That's a major issue we always have. Almost all of the group are attending AGU, becoming ill afterwards. So in all our time series, we have this big December data gap. But we were able to go out early in January, and we had a major event in January 2016, which resulted in a 100-year return flood. So during such wet conditions, we have now really depleted soil water samples. We couldn't sample in February, the soil was frozen, but then when we are now moving into spring, that's March, and then April, we are moving back into a more enriched signature, and then also you see now in May, our PET rate, so the inner shading of the circle is starting to increase. And then during the summer months, our soil water signature becomes more and more enriched, again moving away from the fractionation, and even still in September 2016, so this sample was just taken a few weeks ago, we have still a highly evaporative signal. We can then use these isotope data to estimate the so-called line condition axis. And the line condition axis gives you an idea about the deviation from the local meteoric water line. And as more negative your line condition axis is, as more evaporative fractionation occurs. So here now we see the data averaged over these first 20 centimeters, and we use the same color codes for the different seasons as I was just talking you through on the previous slide. And we found a very strong negative relationship, so when our ET rates increase, our average LC signal becomes more negative, so we have more evaporative fractionation. Indeed, we think we can think about this soil water isotopes undergoing a systematic hysteresis in their fractionation signal, linking the LC signal with the monthly evaporation rates. So when we look at this, starting in winter, we have a really or no evaporative signal, so our LC signal is 0. When we move then towards spring and summer in our sampling, our catchment becomes drier and drier, our PET rates increase, and also our LC signal becomes more and more negative. And then in autumn time, we are moving back to the original signal. And of course, such hysteresis in our soil water isotope is really important in terms of that the water the trees are accessing seems to be seasonally changing. And it's also important when we conduct ecohydrological separation studies, we need to consider when we are conducting the sampling. And we can then use these isotope data and incorporating them into these 1D coupled flow tracer models. And again, this work is still done by Matthias. So this plot scale model uses precipitation as an input, evaporation as an output, and hence accounts for the changes in the soil water isotopic signatures with steps. Again, it's quite a complicated figure, so let me just briefly talk you through. So on the upper profile, we have the estimated LC signal values extrapolated over time and with soil depths, with very negative values indicating high evaporative fractionation shown in red, and 0 values or not so negative values in blue, low or no evaporative fractionation. And then the lower panel shows in these shaded plots the recharge towards groundwater at 50 centimeter depths, and then also the LC signal in this recharge developing over time. And the box plots are summarizing the distribution in the LC signal over the entire time period. So such modeling helps us, for example, to really investigate how this evaporative signal develops over time. So starting the end of winter or spring, we have a low evaporative signal. Then over summer, our temperatures increase, PET rates are higher, so our LC signal increases, particularly in the upper horizon. And then when we have an increase in rainfall, we have a slow flushing down of the LC signal down the soil profile, further mixing occurs. And then at one point, already about 40 centimeter depth, the mixing is complete and we can't really identify this LC signal anymore. What I find interesting as well is that we can see when we are moving back into winter, our LC signal in the upper horizon is becoming less again, so more moving towards 0, while we still have a higher evaporative signal in the deeper horizons. Looking at the recharge, we have in winter, spring, high recharge fluxes towards the groundwater. They are moving this LC signal downwards more negative. Then in summer, we don't have much recharge. This LC signal plateaus out. And then again in autumn, winter, when our recharge fluxes increase again, we have a further downwards movement of these negative LC signal. So very useful tools, but again, I have to emphasize this work is very much in progress. For this initial modeling, Matthias used previous data, but we haven't managed yet to really incorporate our high temporal and spatial data into such modeling. And another work in progress is led by one of my other postdocs, Sylvain Kappe, who is really trying now to integrate all these findings, all these data into a catchment scale tracer-aided ecohydrological modeling. And for this, we use a model which was developed by Marco Maneta, which is called ECHO, to really advance our previous STARR predictions, incorporating this role of vegetation much more. The first step is to adapt this model for these northern region and the processes which are important in these northern region. But crucially also, Sylvain will incorporate a stable isotope component into the model. So to really understand the total storage processes, mixing processes, and the role of vegetation in an integrated way. So as I said at the beginning, across these 6 VeWa sites, we are conducting the same sampling strategies, and the plan is to apply all the models across all these 6 sites. So on my final slide, I would like to show you a stable isotope data set, which I'm extremely pleased about and also extremely proud of. As I said, we conducted the same sampling strategies for all these sites, and these data are just coming in. Most of the soil water samples are analyzed now. Most of the xylem waters, particularly from spring and summer this year, are still in the lab and being analyzed. But we get already first insight into these processes. So this time the color code shows you the sampling for the different sites, and we see how nicely the soil water samples plot along this gradient with very depleted samples at our cold site, the Moss Creek and the Wolf Creek site. And then Doerthe, also here in Canada, is shown in red, much more enriched and very similar actually to our site in Scotland, shown in blue. And all the sites show this fractionation signal during the summer months. Also, the few xylem water samples we have already show some fractionation signal. But please watch the space. I hope by AGU or very soon we will have this entire data set together. And of course, the idea is to investigate whether we see a similar hysteresis signal, which I was talking about for the Scottish site at all these other sites as well. So I hope I could, in my presentation, show you how important I think these integrated cross-scale approaches are to really advance our ecohydrological process understanding in these changing northern regions. We also think that these coupled flow tracer models are extremely useful, not just to stimulate the stream isotope response, but also the soil isotope response. But we also acknowledge we really need now to go beyond that and finding ways to incorporate the influence of vegetation. We found through our more detailed empirical studies, again, that in our system we have a rapid uptake of these evaporative waters from these upper soil horizons during summer. And also importantly, we have seen this seasonal hysteresis in the fractionation signal in the soil water, which might influence the water the plants are accessing. And it's very important to consider when we conduct ecohydrological separation studies. And I think breakthroughs in ecohydrology are possible through such coupled tracer-aided plot and catchment scale ecohydrological models. And with this, I really would like to thank you for your attention.
Thank you, Doerthe. I think we have time for questions. Yeah, Alan.
Thank you. A very nice presentation. Alan Barr from Environment Canada. The question I have is really about network design, and it's a chicken and egg type question. Does the data come first and precede the models, or do the models come first and drive the data collection and the network design? Is that clear?
Yes, it's clear. I mean, I'm probably I know exactly what you mean and how difficult it is. I think I'm an advocate of starting with getting some data, so looking at data first, but incorporating this into your models, but then not viewing the models as your endpoint, using them to inform your sampling strategies, and then going back into the field, renewing your data input, improve the parameterization of your models, and probably going back in the field. So I do think it's a very linked cycle and can't be viewed that one or the other can be the endpoint.
Thank you.
Yeah, Ali. Maybe use the mic if you can. Yeah.
Thanks, Doerthe, for this talk. And this is impressive works, the models you develop. I personally ignored many of these processes that this model and your work consider in one platform. And also you are considering tracer-based information to improve your model calibration and validation, and it's very impressive. My question is about the isotopic signatures of the precipitation and stream flow that you've shown. That stream flow isotopic signature have been very dump, as you mentioned, almost constant. Do you think this type of stream signatures can tell us lots of things about what's going on within the system with this sort of dumped stream flow isotopic signatures? And also, did you do any sensitivity analysis to see how the response of this tracer-based approach is sensitive to the parameters of the to the parameters which are related to the isotopic signatures approach, such as, for example, if you are determining transit time distribution, you have to first priori determine a function and calibrate the shape parameters of that function to determine what's transit time. So all these things can be sensitive to the stream flow, and that stream flow is very dumped. Did you do any sensitivity analysis to see how sensitive are these parameters to the stream signature?
I mean, you are absolutely correct. The way we plotted this here, it looked very damped. It is very damped in comparison to the precipitation input. However, how any isotope person or a scientist would plot these data is to have a different scale in your stream water response and in your precipitation input. So within the precipitation, when we zoom in, we actually see a lot of variability. We can identify snow melt effects. We can see fractionation processes. We see direct linkages with precipitation events. So I think there is a massive information content in the stream water output signals, which were plotted very compressed, but in fact, particularly at our Scottish site, are actually quite highly variable. I can show you other systems where we have a strong groundwater dominance. I mean, then it is almost a flat line. And of course, then you have to ask this question, are deuterium and oxygen still the right stable isotopes to use in these systems? So in our system, they are appropriate because just the age of our water we are investigating. But I think this has to be always viewed with caution in other really groundwater-dominated systems, whether you need other stable isotopes. So I think this was the first part of your question. But then the last part of your question, if you could just did I answer this yet in terms of the...
Did you do any sensitivity analysis to see how this tracer-based, how the results you obtain to inform the behavior of the system is sensitive to a stream signature?
Yes, I mean, we have done a lot of sensitivity analysis in terms of the different parameters, the alpha parameter, the beta parameter, and how sensitive they are on the stream water. I mean, but I think there has been not just our group, a lot of group have done work on getting exactly, as you say, an idea about what is the sensitive parameter, but again, it's system dependent.
Okay.
Yeah.
Thanks. Let's keep going. We'll come back to you, Ali. Other questions? I might just have you go back one slide, ask you an isotope question, prompt maybe Habame or Marcel or others to ask a question. So I guess what you're saying here then, you're seeing this kind of ecohydrological separation, if you like, with the xylem water data, in that they're following a slope similar to the evaporated soil water signature for each of these different sites. Is that the general message here?
No, I think the general message is that so far we don't see any evidence for ecohydrological separation.
And what do you base that on?
That kind of when we do have the deviations, for example, we see is still the fractionation signal, the evaporation signal. I think we need to wait until we really have analyzed the spring samples, the summer samples, whether we can see the direct linkages with the soil water. So the deviations we see at the moment and the plotting we think is explained by the fractionation in the soil water just in these upper soil horizons.
But I guess if you overlaid the right plot over the left plot, the green would follow the green, the red would follow the red, generally, wouldn't it?
Yes, but then I would ask you, so where do you see then the evidence of the ecohydrological separation at these plots? I wouldn't go so far yet at this stage. I would like to wait the other samples.
Yeah.
In terms of really linking, because important is we have mixed here different seasons. They are mixed up. So different. So the thing is, we need to really put the timing of the soil water and the vegetation sampling together. I think that's important. I would think that's too premature to be able to do this yet.
Okay. Howard.
So I really enjoyed the talk. Thank you very much. And thank you for coming to see us. My question's about the implications for hydrological model structures. And for a long time, for many decades, we've worked with these simple box-type structures, and we know that we've got a challenge because we calibrate them to represent a dynamic signal that represents, if you like, a pressure wave through the system. And yet, those models sort of fall over when we think about them trying to represent the transit processes of molecules of water. So I'm interested in the implications of the isotopes for model structure. And I noticed in the models that you put up, you'd actually added an extra element of maybe inactive storage in each of your components. That seems to be something that you found necessary. And maybe you could comment on that. And I guess the follow-up is that doubles the complexity of your model in terms of parameterization. And then you have a real challenge in identifying those parameters from the available data. So maybe you could comment on those 2 points.
Yes, I mean, did you say I didn't hear you correctly? You said about the active storage or the component on the active storage?
I said that it seemed from your model that you'd introduced an extra component, so you had an active storage and a non or lesser active storage.
Exactly, the less active storage, and that's in a way what the tracers told us we needed to incorporate this large passive storage or inactive storage to account for the damping, which we see in the isotope signal. And this was very informative that just focusing the calibration on the discharge response, we wouldn't have needed this, but it was really needed to account for this large volume we needed to incorporate to account for the damping in the signature. You are absolutely correct. Of course, as soon as we start to incorporate stable isotopes as well, we are increasing our parameter sets. But we still always start as low as possible, and even now the model, it's not the last one I was showing, but they are having maybe 7 or 8 parameters to calibrate, despite simulating the discharge and the stable isotope response. So we are still in the limit which is acceptable to us when we talk about LAMP parameters. But this is really this, as you say, finding this balance and incorporating this additional process information and then the tracer information as well. But we find the advantage of incorporating the tracer information outweighs the additional uncertainty through additional parameters.
It would be interesting to look at some alternative, more process-based models and see whether they have the same hang-ups as the conventional conceptual models.
That's exactly the plan now with this model I was talking on my last slide. So that's really a distributed model, physically based. I can't even remember how many parameter sets. And then, as you say, testing this.
Thanks very much.
Yeah.
Okay, we're just at 4:30. Any final quick question before we wrap up? All right, so I'd like to have everyone join me in a round of applause for Doerthe. And before we do, we're going to go to Alexander's for a beer after this, so please join us if you'd like. Thank you again, Doerthe.